Transcriptional control of gene expression is an important control mechanism in eukaryotic cells. And this transcriptional control is going to determine really whether the RNA is produced at all. So this determines if RNA is produced. This is something that would certainly be taking place in the nucleus, because that is where our transcription process would occur. And ultimately what it boils down to is RNA polymerase able to bind the promoter and begin transcription. And if that can happen, then we're going to get our RNA molecule produced at the very end of it. Okay, so let's take a closer look at the promoters in eukaryotic cells and how this initiation of transcription would take place. So here we have a look at a piece of DNA, and what's being shown on here, the blue down at the bottom would be the gene, and the gene is going to be what gets transcribed or copied into RNA later. So this would be located right here, our +1 site, so that would be the transcription initiation site. The part shown here in green is what we refer to as the promoter. And the promoter is going to be very important in actually getting the RNA polymerase to the DNA and to ensure the transcription of this gene at the right level. So when we do start talking about eukaryotic transcriptional control, the promoter is really key to all of this. Now we do have different RNA polymerases involved in transcription in eukaryotic cells, and we're going to focus just on the RNA polymerase II and the genes that RNA polymerase II would actually be transcribing. So in those types of promoters, first off, we tend to have what we call a Tatah box. And the Tatah box is located somewhere here near where the transcription initiation site is going to be. And the Tatah box is really a series of T and A, or thymine and adenine repeats. That's very characteristic of these promoters, and that location, if we just write it here, this is a place where many transcription factors, and we call them TF2s, bind. And so transcription factors 2s are small proteins that are going to help both identify promoters and also recruit RNA polymerase to the promoter area. And that Tatah box is the first place that they bind. So if we talk about that a little bit more, the very first transcription factor that's going to bind is what we call transcription factor 2D. So I'm going to show it right here on top of the Tatah box, and I'll just label it here. This is going to be D, or transcription factor 2D. And once that particular transcription factor binds, then it does recruit additional transcription factors to bind. So there will be a number of other ones that bind, and I'm just going to show a few different ones collected here on top of the Tatah box and in this general area. And then once we do have this collection of transcription factors, then generally we do get recruiting of the RNA polymerase. And RNA polymerase is also going to bind to the promoter, and it's going to bind in this general region. And that right there, now we have assembly of most of the components that we do need for this overall process. At this time, we will generally get phosphorylation of RNA polymerase. So we will see that a phosphate actually gets added. And once the phosphate is added, then that really activates the whole transcription initiation complex. So that is the point where we then do really start to get initiation of this process, and we'll start to see movement of the RNA polymerase down the DNA. Now, in addition to all of this, we can have some other parts to the eukaryotic promoter that help to just make all of this occur just a little bit easier, or facilitate the binding of the transcription factors and also RNA polymerase. So if these are located on the same piece of DNA as the gene, and so usually we're talking about in the promoter sequence, these tend to be what we call cis-acting elements. And these cis-acting elements, these are binding sites for other proteins that are going to help to recruit machinery to the DNA. And these other proteins that will be coming and binding to these cis-acting elements, we tend to call them transcriptional activators. That would be if they're going to actually increase transcription, but then we can also have ones that decrease it. And so those would be called repressors. And so repressors will be kind of blocking or inhibiting this whole process. So if we had a protein that's binding to those cis-acting elements and it's decreasing the transcription, we call it a repressor. If it's increasing it, we call it an activator. So the cis-acting elements are located very near the promoter, or actually within the promoter sequence itself. And that's going to help those activators and those repressors to bind in response to some environmental stimuli or some message that they get inside the cell. There are also regions that are much more distant, and those we see up here, these are called enhancers. And so enhancers are also sequences within the DNA. So here's several of them. And again, the key to these is that they are located distantly from the promoter. And so with these, they're still binding sites. They are for transcriptional activators and repressors to bind to. But the key with these is that once there is binding to them, then we're going to have to get that to actually really come into contact with that whole promoter region. So what's going to happen with the enhancers is that they're going to be bound by transcription factors, transcriptional activators in many cases. And when those bind, then we also have proteins that are going to bind here and help us to really bend the whole DNA molecule. So these proteins, what I'm showing in yellow here, would be what we call DNA bending proteins. And the DNA bending proteins are going to do just like it sounds. They're going to be actually bending the DNA molecule. And when they bend the DNA molecule, it's really going to close this gap that we see right here. And once that gap is closed, we're going to have contact between those enhancer elements with the activators bound to them, and then also the transcription initiation complex with the TF2s and the RNA polymerase. And that is going to, again, facilitate the beginning of that transcription process. And once we get the process started, then ultimately we should get the RNA produced at the very end. We can also have transcriptional repressors bind to those enhancer elements. So those enhancer elements, if they were bound by repressors, we would get the same bending of the DNA and the contact, but in this case, it's going to actually stop or prevent transcription initiation from occurring.